The first thing I always tell people is if you're going to work in founding and building a new company, it's going to be hard, and make sure that it's worth it. Make sure you're working on a problem that's really going to solve something major and unlock something that really isn't possible.
Welcome to the Labs Report. I'm Avak Kahvejian, president of Flagship Labs and general partner at Flagship Pioneering. Last week on our Substack, the Labs Report, we published a deep dive on Abiologics. Today, we're talking to Mike Hamill, origination partner at Flagship Labs, who was part of the founding team. Mike, welcome.
Thanks for having me, Avak.
So you came to Flagship from Flagship founded Excella before you started working on Abiologics, and you were leading research teams there. What was new for you as you came to start doing origination here at Flagship?
When I started at Excella, it was just at the beginning of my career. So I really grew up in the Flagship ecosystem, and I spent a while there. So I really got to understand the operations of Flagship Labs like companies and the principles that Flagship is really built around. I was really excited to join Flagship itself and go on that journey again and work on new companies. I think what was really consistent for me was I got a chance to work on cool science again. I got a chance to operationalize it, work with young scientists on cool new ideas. Some of the challenges, though, were there's just so much you can do at Flagship. There's so many smart people, so many ideas that we can consider with the capabilities we have, and so many experts that we can tap into. It's in a way like pretty overwhelming when you first set into it, and it's something I talk to people about now as they join. So you have to balance it with focus, and fortunately that was something I think we were able to do pretty well in the early protoco journey at Abiologics. So nobody understands the origin of Abiologics better than you do, Avak. When I joined, it was FL83, and there was one employee in the company. We hadn't even synthesized our first peptide yet. But tell me about what happened before I joined.
Yeah, so we, as we always do at Flagship, like to ask what if questions. And oftentimes those what if questions are inspired by things we're doing at the frontier at another platform company. And in this case, Generate was one of the inspirations for this. At Generate, we were doing in silico protein design or protein imagination using AI. And we were thinking about creating traditional therapeutics with that capability. And fast forward to today, that's what the company is actually doing. Peptides, enzymes, antibodies, and beyond. But at the same time, we said, what if now that we have in silico generative capabilities, could we start designing or imagining proteins with different building blocks? Things that natural systems can't use to pan and scan or to prototype. And only a computer can start to put these things together, see if they work well together, see if we can make it, and so on and so forth. So that's when we thought about using mirror image amino acids. So amino acids that are left-handed and are the standard amino acids, and that's what all of nature uses today, as you know. And the D-amino acid world is atomically identical, so all the atoms are the same, and fundamentally they look and feel like traditional amino acids, but they are mirrored images of those amino acids in three-dimensional space. It's akin to having a left hand and a right hand, and trying to fit a left hand into a right-handed glove doesn't work, just like trying to fit a right-handed into a left-handed glove doesn't work. So we thought, what if we gave the computer the mirror image building blocks? Could it fold those little building blocks into protein-like structures? And could we get those protein-like structures to interact or bind to traditional targets in biology? And the only way we could do that is using in silico methodologies. We couldn't give an organism these building blocks. And that was really the origin of all of this. So two things had to be true. The AI had to be able to tolerate those things and imagine interactions. Then we had to make them. We had to actually put those building blocks together in the real world to test whether that works and to give the computer the answer so that it couldn't get better and better at doing what it is doing. That was essentially the origin. I think you came around that time where we had just basically put the idea together and had convinced ourselves that the algorithms can indeed maybe conjure up some of these imaginary proteins, and that we had talked to some experts that said indeed we could probably string these building blocks together.
I had been working in amino acid metabolism at Excella for the past decade, but my PhD was in structural biology and biochemistry. So I came to the table from really a protein sciences mindset. And so to me, seeing the story, and it was Nick Plugis who walked me through it the first time, I think it was like 5 or 6 slides into the story where there were images of all sorts of different types of protein modalities that were kind of painted as imaginary things we could build. And in my mind, I started looking at like the antibody that was on the page and thinking about, sure, you could conjugate a linker to it, you could have a payload, but you could also put pharmacophores on the binding site to make it more potent and selective. You could completely change the backbone to be able to make it resistant to proteases and non-immunogenic, as we had learned and hypothesized from the D space. And it just seemed so profoundly powerful to me, knowing at the same time that that was an extreme vision. We were never going to synthesize full-length antibodies, but it was really useful for me to kind of wrap my head around all that could have been possible if we built this thing.
So when we present Abiologics today, some might say this looks very obvious, super easy. Why don't you maybe unpack a little bit of that? What were some of the challenges of getting us to this point?
Yeah, and I think, I think good ideas also have really strong anchoring in history too. There was a lot of D-amino acid history. D-amino acids were incorporated in peptide synthesis shortly after solid phase was invented. They ended up in drugs in the 70s. And so the concept of stabilizing peptides with D-amino acids had been around, but bringing it to bear at scale against any target, the technologies that existed really weren't there yet. It really didn't fully unlock the potential of the space. And so as we sat down and we looked at the AI landscape, as you described from Generate, we knew that there were really powerful tools in the protein world that were available, but we also knew that none of them worked in the D-amino acid world. That wasn't what they were trained on. It wasn't what they were hard-coded to do. And at the same time, we knew that nature was not going to make these things. Nature wasn't going to be able to take D-amino acids for, 30, 40, 50 amino acids long and string them together in at all, if not in meaningful amounts. And so we had to solve a chemistry problem. And synthesizing proteins with chemistry is very different from how nature does it. Nature has, 4 billion years of evolution the ribosome has to get really efficient at the chemistry that it uses to string amino acids along. It's infinitely efficient. In the chemistry world, 99%, 98% is great efficient chemistry. But if you're making something that's 100 amino acids long and you're adding an amino acid and then removing protecting groups, and so each step is multiple reactions, that compounds over time, and it's really, really hard to synthesize long protein length things. And so those were really the two challenges that we had in front of us, and what we've had the team working on the last few years and cracked wide open. So yeah, on the protein side, the field evolved as we were building the company. So we've got all sorts of tools available to us now that we've figured out how to apply them in the D space to do design. And then simultaneously, the chemistry had to happen as well in order to enable that computation, because we could have tried all the things in silico in the world that we could imagine. If we didn't have feedback from the lab on binding, yes, but also folding, solubility, the output of the synthesis, then it would have been really hard. And so as we went, we really moved that along. And, we started with goals hoping we could make hundreds of 20 MERS, then hundreds of 30 MERS, 40 MERS, 50 MERS. And at this point, we can regularly go up to 60 amino acids long, which is really important because that's when these sequences become proteins and the biologics nature really comes to bear.
So why don't you give us a little bit of color into what were those hypotheses we had, and then sort of the experiments we did to validate or test whether those properties came true.
Yeah, and I actually think this is also an interesting approach that we took at Abiologics from just a company build standpoint, because we were working on hard problems like the AI generative design of this whole other universe of protein space and the synthesis, but we always had a north star of those two really hard problems being worth solving because of what they would unlock in terms of the properties that they could have. And so we didn't really wait for those tools to be built to start asking the questions that you just mentioned. We started right off the bat ordering libraries of L peptides and proteins, and then the same exact sequences out of the D amino acids, and testing how they behaved in animals for important things that you'd want out of drugs: stability, PK profile, immunogenicity, and of course, this, like I said before, these D amino acids had been around for decades. There were little pieces of literature that pointed to these properties, but you really want to see it for yourself. And you want to see it not just apply to one unique place, but systematically across the modality. And that's exactly what we ended up seeing. I think one of my favorite experiments that we did, even early in Protoco, was when we took two of these libraries and we wanted to understand the PK profile, and we injected them into some mice, L amino acids in one, D amino acids in the other. And of course, we looked at them in the plasma and we did the normal things that you would do with peptides. But we also had kind of an odd idea that, well, if they're really stable and they end up getting cleared out of the body, they should end up in the urine of the animals. And so we put the mice in metabolic cages and collected the urine at the end of the experiment. And sure enough, when we looked in the droplet, we saw, next to none, very trace of maybe one of the L peptides we had put in there, but we saw every single one of the D peptides in there and in meaningful amounts. And that, to me, was one of the most exciting and kind of like profound results, because it was just so different. And it was something that you normally wouldn't have applied in the peptide space, because you'd expect them to degrade over time, even if they had been stabilized with one or two non-canonical amino acids.
Yeah, and then we started to push the boundaries, right? So you mentioned PK immunogenicity. We didn't talk a lot about that, but that also is a striking difference. And then we started to push the boundaries into tissue penetration so that we can look for therapeutically relevant kind of activities or properties, right? So some of the data we've put on our Substack is to really showcase and bring to life the visuals of how these things go beyond the vasculature and penetrate deeply into tissues. What are the implications in your view of all of that? And what are we trying to do with those data?
Yeah, and I think, to go back to the stability and the immunogenicity, at the time with all the protein design work that was happening, there was a lot of discussion around how these tools can help the biologic space. And one of the dimensions of that was, when we're working on different proteins, you're not just optimizing for potency, but you're optimizing for multiple objectives. Multi-objective optimization of proteins was something that was discussed a lot at the time. And of those objectives were things like stability and immunogenicity. And so we always thought it was really interesting how, in this case, you took a few of those dimensions and just fully took them off the table with the data we were seeing on stability and immunogenicity, because of the nature of these D peptides not getting degraded and then, not being recognized by the immune system. And then the moment you're mentioning, so thinking about the tumor penetration data that we're sharing, that was really the first time that the platform converged, where we had the AI and the synthesis and a syntein that we had created ourselves in-house for the first time. And it was an early prototype of one to an important target in oncology. And we combined that with, what we had seen with the protease stability and the biodistribution of these things when we were studying them as tools. And for the first time, we could draw a comparison against a biologic itself. In this case, an antibody that bound to the same target. And the data we're sharing is when you look at a centine that binds to that target in a mouse where it's had a tumor xenograft, and how the antibody compares to it. And that's the manifestation of what the centine is enabling. Is the centine paints the tumor. You see the red dye, which is the dye that we conjugate to the centine so that you can see it. Same thing with the antibody, and you can see that the centine is everywhere and that it persists for many days. Whereas the antibody is really stuck to the vasculature because of its size and also because it gets degraded. And so if you put that in the context of a drug for oncology, where you're looking to deliver a toxin or a radioligand, for example, it's all about getting maximum exposure to those cancer cells, penetrating and getting to every single cell that you possibly can, and being able to hang out in the tumor as well, to be able to kill for as long as you can. And the Centenes in that experiment just showed us that there are potentially better ways to go at things like solid tumors, and that this technology was giving us an opportunity to be able to go further and maybe get into different cancers that were previously not something you could think about with a more limited modality. This was my first foray into AI from a company building standpoint. Of course, I've got a few others and I aspire to have many more in the future. But it wasn't for you, Avak. You had already started Generate, you'd already started Celerity. What is it that you've learned through being, one of the largest contributors of AI companies to this ecosystem?
Like you said in the beginning, we embark on these things and we're not sure how hard or easy it's going to be. What and in the field of AI, people hear AI and they think it's now capable of doing anything. It's just about applying it to the right things. But in our field, I think in the field of biology, AI is not just the use of LLMs. It's not just the use of a chatbot. We're actually building new AI models. And those models need data that doesn't exist, or they need to see experiments and the results of those experiments to be retrained and perfected. You need to go through a reinforcement learning loop to make those AI models work. And we've seen that time and time again. It's not as easy as it sounds to just use an AI to generate things or use an AI to analyze things and then move forward from there to create the ideas that were generated and go to the next step. In fact, you need that design make test loop. That's why it's called that. And each time you don't know how hard is it going to be to train the model. You don't know how much it's going to hallucinate and create useless things and how often it's going to create useful things. And you have to climb that curve. But then that means designing an experimental setup as well that's capable of generating the right kind of data, the right amount of data in the right time frame. So at Generate, we learned that we could do it with L proteins, and it took a long time. At Celerity, we trained on single cell transcriptomic data. That took a long time. And now at Abiologics, we were generating data no one has ever generated before. The interface between D peptides that really never, many of which never existed, interfacing with L targets, those data don't exist. You can't just train on publicly available data and have the algorithm design new ones for you. So we had to basically create those data from scratch. And as you can imagine, and you lived it, we made a whole bunch that didn't work. So those were a lot of negative data, which is useful, but you need some positive data. And so those are some of the challenges you run into when you just say AI in biology. It's not as straightforward as one would think, depending on what you're trying to do. And so when you're trying to create a new modality with AI, that means you're creating a new AI as well. You're not just creating the new modality. So what advice do you have after having gone through this journey with us, with me on this, to someone starting off as a scientist entrepreneur in this kind of pioneering platform building realm, not just the simple entrepreneurial realm, but doing stuff that's in uncharted territory?
So the first thing I always tell people is if you're going to work in founding and building a new company, it's going to be hard, and make sure that it's worth it. Make sure you're working on a problem that's really going to solve something major and unlock something that really isn't possible in other ways. And in this case, obviously, the vision that we crafted as we were building the company fit exactly that bill. And then the other thing is building off of what you said in terms of the data that we've generated, working through all of those hard problems, but really focusing on data generation, reacting to data, learning from the data. Now we can look back and see, over 20,000 of these things that we've built and all of the data that we've captured and are able to reflect back on. And all of the learnings that come from that are something that we leverage every single day. So capturing all of that and being, really cognizant of what can be done through retrospective of a great platform that's generated, yes, plenty of bad data, but also good data and interesting data that can be interrogated. And then finally, I think just, now as we're building biotech companies, we really want to think about the pipelines as early as possible. Our two main programs that we're working on in oncology and in metabolism are really what we built the platform around. And that's really exciting. And it really is only possible as well because these are projects that we're able to talk about with the Pioneering Medicines team early in the protocol and be able to decide or figure out that these are areas where a platform as we build it could have a huge impact. And so having that team and that expertise within Flagship and being able to make those early decisions. So now we're not going back and saying, oh, all right, now we'll apply this to a pipeline project that we believe in. Being able to have built the platform and ended with it just gives us a major head start and a really exciting next couple of years, I hope.
Thanks for giving us a sneak peek behind the thinking process, the journey for generating the data. All of the data we talked about is on our Substack. Thank you very much for joining us.